Hello, everyone. All right, can people in the room hear me? Is this microphone great? And can people on Zoom hear me? All right, we have a lovely crowd on Zoom, and it's growing, but I think we're gonna jump in and start. Hi, everyone. Welcome. My name is Erica Reinfeld. I am the Outreach and Communications Manager here at the Koch Institute. One of the distinct pleasures of my job is to produce every year, with the help of a lot of people, the annual Image Awards exhibition. I am so excited to be welcoming us for our very first hybrid public program around the Image Awards. You'll notice that I'm taking my mask off. This is consistent with MIT policy. Some of our presenters will be unmasking. Some of them may leave them on. Our audience is masked, so we are staying safe and trying to include everyone we have. So it is again my pleasure to welcome all of you to the opening of the 2022 Image Awards. I will be your guide for the evening, but I'm going to try not to talk too much because what you're really here to see and hear are the images and the stories behind them. I have in the room with me the people who created these images and many of the people who supported them, and we have lots of moral support happening on Zoom as well. So we will include everyone. So before we jump into the presentations, basically we've got 10 images and 1 hour to hear all about them. So our presenters will be presenting a series of lightning talks. But before we do that, I have a few thank yous that I want to give, assuming that my clicker works here. It worked when I practiced, so we know what that means. There we go. All right. So as I said, this is our Image Awards exhibition. These are the winners of our 12th annual Image Awards competition. This year we had about 125 images submitted, and our esteemed panel of judges, at least one of whom is in the room, and I think there's some more on Zoom, helped recommend the 10 images that you're going to see tonight. And then I also want to thank Charles B. and Ann L. Johnson for the generous gift of establishing the Philip Alden Russell Endowed Gallery Fund, which provides ongoing support for our exhibit renewal. So we are very grateful for all the people who helped put this exhibit together. And I think in a little bit, we'll hear from the images, and we'll also hear from our new director of the Koch Institute with some closing reflections. So with that, I think we're going to bounce all the way to the far end of the gallery to talk with actually one of the labs of their director's suite mates, the Belcher Lab. And alumnus Eric Lenhart is going to talk to us about copper seashells.
Thank you. Today, I'm pleased to present to you an image that I captured of a catalyst. In chemistry, as in the real world, a catalyst is something that accelerates change. The change that my collaborator, Dr. Will Records, my advisor, Professor Angela Belcher, and I want to accelerate is a transformation of the way we make our fuels and plastics. You see, so much of our modern environment is built on the products of ancient photosynthesis. Over eons of time, water, sunlight, and carbon dioxide were combined via photosynthesis to make sugars that make leaves and bark and trunks. Over eons more time, those ancient leaves and bark and trunks were buried under intense heat and pressure until they became the fossil fuels that we use today. They are critical not just for transportation, but to heat our homes, power our grid, and act as the raw material for everything from polyester fabrics to medical devices. Yet when we use these fossil fuels and we use the energy of ancient sunlight, we also release the ancient carbon as carbon dioxide into our atmosphere. But what if, like nature, we could pull the carbon that we need from the air and not from the ground? We would need special catalysts to do this that could take water, carbon dioxide, and electricity to make the fuels and plastics that we need. We would have to go faster than nature does. We don't have eons, but biology can still help us solve this problem. Biology is an expert at making high-performance materials. Think of seashells, which are 98% the same as chalk. That is, they're 98% calcium carbonate. Chalk snaps easily, it crumbles in your fingers, but seashells have a toughness, a rigidity, even a beauty, and that comes from an extra 2% content that is protein. And the protein carefully arranges those calcium carbonate atoms into a crystalline structure. Biology does this everywhere. Think of the oxygen in your blood that is carried by hemoglobin, which is a complex of iron and protein. In our research, we use biology to inspire and guide the creation of high-performance materials like catalysts through the blending of proteins and minerals. The image before you now is a bio-inspired catalyst built to help produce fuels and plastics from water, carbon dioxide, and electricity. Each of these pillars is made of copper metal, carefully arranged atom by atom to help us investigate the best way to convert carbon dioxide into the products we need. The pillars are about 10,000 times larger here than they are in real life. We made this catalyst and many others like it using a process called electrodeposition, or sometimes electroplating, which is the method by which the nickel core of a penny gets coated with copper. We made these catalysts from a bath of dissolved copper, using electricity to drive copper ions onto the surface of our catalyst and slowly build up layers and layers of metal. But we introduced a biological twist, which is that we added engineered proteins to the bath that changed the way that the copper formed its structure. This is not unlike the way that protein changes the structure of chalk as it is built into seashells. The picture on the right shows what our catalyst looks like at the laboratory scale with the naked eye. With this catalyst, we hope to answer questions about how catalyst structure can dictate which fuels and plastics get made from carbon dioxide and water. The ultimate goal is to replace fossil fuel inputs at refineries with carbon that's harvested from the air. And many labs and companies are continuing to pursue scale-up of catalysts for this chemical reaction. I was sitting alone in a room with no windows when I first saw these pillars, but when I did, I immediately thought of all of you. And I took an extra 20 minutes to capture a single high-resolution image that I hoped that I would be able to share with you today. It is my prayer that catalysts like this one will help accelerate our ability to harvest and use carbon dioxide from the air. Science is a team sport, and I would like to thank the many folks who contributed to the work behind this image, especially my advisor, Professor Angela Belcher, my collaborator and lab mate, Dr. Will Records, and also Dr. Nicole Bone of the MIT ISN, who taught me how to use the microscope that captured my image. Many thanks also to Shell, the Sloan Foundation, and MIT Biological Engineering, who helped fund this work. Thank you.
Thank you so much, Eric. I think that was 20 minutes well spent and probably a thesis worth of work as well. Thank you. So our next image that we are going to do, actually, I did want to say that they really do look like seashells. And so I think when we have people walking through the galleries, we're going to try really hard to fuel them. I have really missed the live stage. All right, so our next image is a different kind of pillar. This is also an engineered pillar, and it combines biology and engineering. And I'm going to invite Ning and Anna up to take us on a journey down the astrocyte highways.
Good afternoon, everyone. My name is Ning, and this is Anna, and we're researchers in the Van Vliet group. We're really delighted to be here today to share with you this image, both for our attendees in person as well as those on Zoom, actually including our families who are zooming in from Poland and Hong Kong. Now, what you see in this image, colored in magenta, are human astrocytes, and these are a key supporting cell type in the brain. These astrocytes are extending their many cellular arms, snaking their way around a 3D printed material shown in red that we developed in our lab. Now, to tell you the truth, like most things in research, we actually discovered this quite beautiful cell morphology entirely by accident, because astrocytes aren't even the main cell type we study in our group. But to understand how we got here, I'd like to take you back to the beginning. These astrocytes represent a profound paradigm shift in biology. You see, these cells, even though they're found in the brain, were actually obtained from stem cells derived from human skin tissue. Our ability to derive neural cells in this way is really transforming our fight against neurological diseases. Currently, there are no cures for any neurodegenerative disease, such as Alzheimer's disease or multiple sclerosis. One of the major challenges this field has faced has been the lack of access to human neural cells with which to study disease mechanisms or to screen for drugs. Although animal models have been helpful, oftentimes those findings haven't translated to human patients. In our brains, we have a rich variety of different cell types, including the main functional unit, neurons, and a rich cast of supporting cells called glial cells, including astrocytes, our favorite oligodendrocytes, and microglia. These cell types all interact in richly complex ways, and those interactions have been implicated in various forms of disease. Now, over the past decade, scientists have gained an unprecedented ability to obtain and derive these neural cell types from human stem cells. And in our lab and many others, we use a type of stem cell called human-induced pluripotent stem cells, or iPSCs. These are stem cells typically derived from reprogrammed skin tissue, and they're so named because they can differentiate into a plurality of different specialized cell types. For example, iPSCs can become epithelial cells, or blood cells, or neural cells. And in our lab, we can apply chemical factors to coax these iPSCs towards becoming neural cells. Now that we've attained patient-derived human neural cells like astrocytes and neurons and oligodendrocytes, we can use those cells to probe disease processes. And I'll now hand over to Anna, who will tell you a bit about how we use those cells to study one such disease, multiple sclerosis.
A hallmark of multiple sclerosis is damage of myelin, which you can see here in green color. Myelin is this amazing protective and insulating layer that is wrapped by cells called oligodendrocytes around axons, those long arms of neurons that conduct neuronal signals between the brain and the rest of the body. Damage of myelin interrupts the signal, affecting our vision, mobility, and other vital functions. If we could stimulate oligodendrocytes to repair myelin in the process called remyelination, we could reverse the disease. Unfortunately, today we have no cures for MS and no therapeutics to stimulate this process. At the Van Vliet group at MIT, we've been working toward understanding how the microenvironment in MS drives the disease. Toward this goal, we developed a unique platform called artificial axons. In this platform, we use advanced 3D printing and biomaterials to create mimics of axons. You can see those as those red pillars here in this top and side view. These artificial axons mimic the geometry and mechanical properties of real biological axons, such as those that you can see here in this SEM rendered image. This platform enables us to directly visualize and quantify myelin wrapping by human oligodendrocytes. You can see myelin wrapped around these artificial axons in green. We can also watch myelination live. We can now use this platform to study myelination in various conditions that represent the disease, including inflammatory factors, and also in the presence of other glial cells that Ming introduced to you that can also affect myelination. Astrocyte, a star of our awarded image, is one such cell that can affect myelination. Although we haven't studied astrocytes in the past, we now include it as a part of our system and key player in myelin damage and repair. In this image here, you see an example of human oligodendrocyte wrapping a green myelin around artificial axons in the presence and close interaction with an astrocyte. Our goal is to use artificial axons to discover new therapeutics for myelin stimulation, the repair of myelin in MS. And with this, I would like to thank you and acknowledge our collaborators at MIT and the New York Stem Cell Foundation and funding, and especially thank Erica and her team at Koch Institute for working with us on this image. Thank you very much.
These are for you. And Eric, I have one for you. Apparently, I forgot this was an awards ceremony, but all right. I am loving the teamwork that's on display, both the cells themselves and also the people behind these images. All right, our next image also looks at cell interactions in a very different way. And I have to say, this is probably the most insulting title we've ever given an image. So Jonathan, if you want to come up and talk about what a dump your image is.
Okay, thanks Erica for the introduction. So I'm Jonathan, I'm a graduate student in Adam Martin's lab over at Building 68, and I'm going to talk to you today about eggs. basically. So egg cells are enormous, at least as cells go. So I'm going to show you a few cell types here, and these are all to scale, starting from, come on, a red blood cell and a skin cell, which decided to show up at once, and then a human egg cell. So this is already much, much larger than a skin cell. And then because our lab works on fruit flies primarily, I'm going to show you fruit fly egg cell next. So these are even huger. And one of the obvious questions that comes up here is how these cells actually get this large and why they don't stay at the size of skin cell, for example. And they need to be this big because egg cells are going to go on to support early embryonic development. So they need all kinds of the goodies that cells need to survive, or that organisms need to survive. So we set out to look at basically the mechanisms for fluid transport that helps these eggs grow. And fruit flies are really cool about this because for fruit flies, it takes a village to raise the egg. So typically, or what always happens is you have one egg cell, shown here in magenta, connected to 15 support cells in this tree-like structure. And there's a whole lot of different choreographed steps that go on here, but towards the end of egg development in a process that's called nurse cell dumping, hence the title, very rapidly over the span of about 90 minutes, these 15 support cells transfer almost all of their contents into the egg cell, basically shriveling up and dying so the egg cell can survive. And what this looks like live using a membrane marker here, I'll show you in a second. And I want to point out two things as this is going. So first, these cells begin shrinking, and they do so without really changing their shape. And then about now, you can see the cells start to wiggle. So these are pretty heavy cell contractions mediated by proteins exerting force locally. So we wanted to determine kind of the dynamics of this dumping process. So in what order and what direction and at what speed the fluid moves. And to do so, we collaborated with Jorn Dunkel's group and MIT Mathematics to try to model this as well. And kind of as a side bit of this, we noticed there's this protein called PCNA that normally exists in the nucleus. You can see it here as these cyan balls. And it actually gives us a hint to when the cells transition from the shrinking to this wiggling type of fluid transport. So this is another movie, and I want to point out, you can already see it in this cell, that the PCNA signal has gone from the nucleus to the cytoplasm, and in the same order that it does that, with roughly a 20 to 30 minute delay, the waves begin in the cells and they start deforming. So this protein's kind of neat because whether or not it's directly causative or anything here, we're still not sure. It does give us a sort of timer to let us know which waves are going, or which cells are going to begin waving and roughly when. So while imaging this one day, I had 4 of these that were relatively near together, and I just kind of pushed them around in the dish to get. Yeah. Nope. There we go. Push them around on the dish to get them arranged like this. And something I think is really fun about this image is knowing what I told you about PCNA, you can tell the age of these egg chambers and how far each one of them is into the dumping process based on whether the PCNA is entirely in the nucleus, like the youngest one at the top, or mostly out of the nucleus, like the oldest one at the bottom. Something I think is cool about this process, besides just this being an interesting way that biology gets things done, is that when we were collaborating with Jorn Dunkel's group, it turns out the fluid flow, at least in part of this process, is very nicely described by a fairly simple physical law. And since physical laws are universal, it suggests that this type of fluid transport could likely be seen in other organisms. And in fact, mouse oocyte development seems to do something at least similar involving fluid transport between cells. Once PowerPoint decides to cooperate, I'd like to thank all of my collaborators here. There we go. Okay, so first of all, Yasmin, she took most of the video, well, both of the videos that I showed you and a lot of the images for this project, and she also trained me to work on the system. And then Adam, my PI, and Jorn and Nico, who did the theory and simulation for this project. And then everybody else in both groups for their wonderful feedback and also our funding sources. So, thank you.
That was excellent work. Look, guys, I'm always going to go for the low-hanging fruit fly jokes. All right. So I'm hearing on Zoom that our presenter, that our Zoom audience is having a little bit of trouble hearing if we step too far back from the podium. So if my presenters could try to casually lean into the microphone, that would be helpful. All right. So we're going to switch gears and move to an engineering project at the other end of the gallery here. This is Nutrient Galaxy. It looks like a whole new world, and Rhoda is going to tell us all about it.
Hey, hello everyone. My name is Lindsay Shuanrou Zhang, and I'm a chemical engineering PhD student from the Langerlach Connect group. Today, I will talk about the story behind this image. So, micronutrients are an essential part of our daily foods. However, in many communities around the world, especially in low to middle income countries, local food products are lack of important micronutrients such as vitamins and minerals. And a chronic lack of these micronutrients is leading to the hidden hunger crisis, which is affecting about 2 billion people globally, which accounts for about 7% of the global healthcare burden. And to combat this global micronutrient deficiency crisis, food fortification has been shown to be a cost-effective solution with wide range coverage, especially for those targeted regions. So specifically, food fortification is defined as a practice of improving nutritional value for those commonly consumed food products by adding vitamins and minerals. And because it is the local food products that are added with micronutrients, the food fortification strategy is independent of the local healthcare system and therefore provides very high program sustainability. However, at this stage, food fortification strategy is not being beneficial for those most marginalized communities because of two main technical barriers, including stabilization of micronutrients under those common cooking and storage conditions, as well as the release of micronutrients from the oral digestion process. And overcoming these barriers are especially important for applications in those marginalized regions because those regions are usually lack of vacuum packaging or cold chains. So in our group, we have developed a polymeric microparticle platform for micronutrient stabilization and delivery. Specifically, we first synthesized a group of novel biodegradable polymers named as polybeta amino ester, PAE, and then we fabricated PAE microparticles encapsulated with micronutrients using the oil-water emulsion method. And here we included a picture of the PAE microparticles with vitamin A and also an SEM image of one single particle, which is part of this colored awarded SEM image. And this kind of SEM images provide a lot of valuable information for us to study the protection mechanisms and also the microscopic properties of these particles. So by encapsulating by encapsulating this micronutrients in the PAE microparticles, our technology has a huge translational potential to overcome those aforementioned barriers. And here we used vitamin A as an exemplary micronutrient. So first, under a mimic of cooking condition, which is boiling in water for 2 hours, our microparticles can provide protection for over 80% of vitamin A in that condition, compared to less than 10% of recovery for the unprotected form of vitamin A. And also, under the accelerated storage condition, over 75% of vitamin A was recovered from the PAE microparticles after 1 month storage, while for the unprotected vitamin A, all of them degraded under the same condition. And this fabulous protection efficiency did not compromise with the rapid release of the system. So after 10-minute treatment of stimulated gastric fluid, which is a mimic of the stomach environment, over 90% of vitamin A was released from the system. So finally, I want to acknowledge my advisors, Anna and Bob, for their support and also our micronutrient team. And I want to give special thanks to David and Abigail from Koch Facilities who helped us with the SM imaging. And finally, special thanks to the Bill and Melinda Gates Foundation and our program manager, Deepika, for their support. Thank you.
Vitamin A+ project. Yeah, on Zoom they can't hear us groan. All right. So this actually kicks off the segment of the presentations that I like to call the digestive session, because our next stop is also the digestive system. So Neil, the pressure is on.
Yeah, the pressure is on. So hello everybody, my name is Neil. I'm going to talk about a picture right over here and I'll talk about a project where we are trying to map the motility in the digestive tract. So in the beginning, let me talk about, hold on a second. Let me talk about what is digestion. So digestion is basically both a chemical and also mechanical process. And when we are talking about the motility, we are talking about the mechanical part of this process. And as you can see from here, you can see about AX making sausage. So after you have your food inside your digestive tract, your digestion is going to grind, process, and then transfer it downward. Well, however, in some situations, when this kind of motion is disrupted and you and your loved ones will feel unwell, it could be scenarios like the food is not coming out, which is we call constipation, or food is coming out in the wrong direction, which is called reflux. And so this is not, so this is not trivial because especially for patients who are having cancers or who are having cancer therapy, those kind of symptoms are very, very common. And so in the clinics, what the physicians are trying to do at here is to use something called manometry. That is basically a catheter of 1 feet long with an array of pressure sensors, and you can insert inside the esophagus and then try to get us an insight of what the motility is in the esophagus. And what we are saying here, if the state of art can only do for the esophagus, what if we build a system that can map the entire GI tract, digestive tract? And so one critical challenge we have to solve at here is to find a material that can transfer the signal from pressure into the electrical signals. And we have been trying to synthesize this material for a long while. But we see a lot of issues like the adhesion with the electrode and also the sensitivities. So long thing short, it's not working very well. And then someday miracle happened. Just like one day, when I was just trying to clean my bench, I was just about to throw some of those kind of foams. And one undergrad student was talking with me, and he said, you know, Neil, actually those kind of foams is actually conductive. And that is because if you are going to buy a piece of very expensive electronic device, and you want to rub it around with some foam that has some level of conductivity, so you can discharge electrons. If you have the electrons accumulating in your device, it could cause severe damage. And so if you're going to see some sort of like pressure applied on this foam, it could have some sort of changes of the electrical reduction. And then we start to find out the solution, and here is the solution. So we screen many different foams, and we realized for particularly this foam, it has the best electrical and mechanical performance. And what you are looking at here is looking at the skeleton of this foam, and the electrons would flow through those skeletons. And if you're going to compress it, you are going to create more connection points inside its structure, and electrons will have more pathways, and you are going to see the reduction of electrical resistance. So by doing that, we are now building a one-way relationship between the pressure and also the electrical resistance. And we are able to build our device, and we are able to conduct animal studies. And now we have a device that can map the motility everywhere in the GI tract, which is kind of very interesting. And so finally, I want to thank Professor Traverso for supervising me. I want to thank another facility core, and I want to appreciate anybody and everybody who has been working with me. Thank you so much.
Thank you. I'm wondering if people are feeling FOMO now. I have certainly learned something from this, which is I need to be very, very careful when I tell people that this Image Awards event is a chance to tell the story behind the image and find out how the sausage is made here. So if you can stomach one more trip to the digestive tract, our next project is Organoid Oncology. And George is going to come and tell us how to make sense of sensitivity. And this image is nothing ostentatious, but it is a very useful tool.
Thanks. So my name is George. I'm a postdoc in the Yeoman's Lab, but like my other collaborators here listed, both Manish and Omer, we're all also clinicians at MGH. And this talk will get into the intersection between those two sort of events. And so the title is Organoid Oncology, Making Sense of Sensitivity. And so that brings us to the beginning where we're talking about stomach cancer. It's the KI, we are trying to treat cancer, and that's some of the sort of tenets and goals that we try to ascribe to. And so unfortunately, there are about 26,000 new cases of stomach cancer diagnosed each year in the United States. And on the left is a histologic image, which is what a pathologist looks at when they look at tissues. And I and Omer are both pathologists. And that's actually a histologic image of normal stomach. And a known risk factor for getting stomach cancer is infection with a bacterium called H. pylori. But there are rarely hereditary causes that can induce stomach cancer. And once you get diagnosed with stomach cancer, often the standard of care involves chemotherapy. And unfortunately, those chemotherapy regimens are not universally standard, and responses can be variable. And so we in the lab are thinking, can we improve this using organoid testing and organoid technology? But what is organoids? What are organoids, and what is organoid technology? And so what we can do is we can dissociate the crypts, so the cells that make up the tissue as shown in the previous image, dissociate them from the underlying tissue and grow them in the lab. And this is an image of those dissociated cells in the crypt-like architecture that they retain. And what we do is we put them in a jello-like material, as shown on the right, and place it in the bottom of a culture well. So we're able to grow it in an incubator and grow it in the lab and study it. And we're also able to overlay it with different media conditions to study things like chemotherapy and response to different stimuli. And so I just want to warn people that maybe sort of like the previous talk, the next image is a little bit graphic, but this one is from the patient that actually the image and the organoids were derived from. And so as you can see on the left, this is actually an endoscopic image of the tumor that the patient had. So this was an endoscope, is like a camera that you advance down somebody's esophagus into their stomach. So this is a picture of their stomach. And it's pretty obvious that there's a large misshapen fungating mass right there in the center. And it's got irregular borders, a lot of funny coloration, and that's in comparison to the smooth, regular kind of aspect of the mucosa on the outer edges. And so what I did, I went with Manish to do this case, and what we were able to do is get biopsies from both the tumor areas seen there, as well as the normal areas. And I brought them back to MIT, and we were able to grow them here as organoids. So on the top is a representative image of tumor organoids, and on the bottom is a representative image of the normal organoids as we grow them in the lab. And so one thing that we knew about this tumor and was specific to this patient was they had a lack of this gene called ATM. And so what we're able to do is study that in the lab by staining for it using a fluorescent staining. And so the presence of that protein is labeled in red. So the normal organoids retain that color, retain that protein as seen on the left, but the tumor actually lacks that protein. And as you can see, there's no red staining. And this was part of the hereditary basis of this patient's specific cancer. And because we identified that and we knew that, we were able to identify specific sensitivities for the tumor based on this knowledge. And so on the left is a line graph showing organoid viability on the y-axis, which is how much the organoids survive. And on the x-axis, as you go left to right, an increasing concentration of a chemotherapy. And the chemotherapy used there in chemotherapy A is a standard chemotherapy used for treating gastric cancer. And so as you can see, the tumor cells actually are a little bit more sensitive in that they start dying earlier when exposed to an increasing concentration of chemotherapy in comparison to the normal. And that's what an oncologist wants to happen. You start killing off the tumor cells before you kill off the normal cells. But because we knew something about the tumor, that the tumor was ATM deficient, we can use a different chemotherapy, chemotherapy D, which shows a huge range where the tumor cells are very sensitive to this particular chemotherapy in comparison to normal. So you can get away with much less dosage of chemo, and you really very limitedly hurt the normal cells. And that's overall the goal that we had with this project. And so this final image, which is slowly coming, is the image that was chosen for the award, and is actually a picture of the normal organoid grown. Okay, and that's the picture of the normal organoid. This is one of the images that I was just struck by, sort of the appearance that it had and sort of the three-dimensionality of it, and that's why I submitted it. So I want to thank Omer, Manish, the entire UMass lab, and of course the patients that consent to participate in research, as well as Erica and the KI Image Awards, and then all of the cores and at the KI, which made this happen.
Thank you. Yes, I always wonder whether we warn people about the samples, and then do we also have to warn people about the graphs? Or I guess the grafts in this case. But thank you. This was a real treatment. It really is a treat to see. Yes, I'm just going to keep going, guys. It really is always a treat to see how this fundamental biology interacts with clinical practice and what's happening. This was a very personalized medicine project. Our next project is also an organoid project, but it is focused a lot more on wider cohorts. So Alan is going to come up and show us how we model disease behavior.
Okay, thank you for the introduction. So, oh, sorry, go back. So today I'm going to briefly introduce my story behind this image award, which is also using an organoid to model the disease behavior. So the disease that I'm mostly focusing on about is prostate cancer, which is a highly prevalent cancer and the second cause of death for men with cancers in the US. And prostate cancer usually originated from the prostate glands, which can also invade it into other normal tissues. And in my research, I'm particularly interested in this phenomenon called cancer aneuploidy, which is featured by gains or loss of certain chromosomes in the cancer cell. And we assess the chromosome gains and losses in prostate cancer by using the data sets from Cancer Genome Atlas program. And as you can see here, some of the chromosomes have a higher levels of gain and losses frequencies than the other ones. So we always have the hypothesis that gains and losses of certain genes on these aneuploid chromosomes by changing their gene expression levels can promote prostate cancer. So to do this research, I've been trying several different 2D cell culture models. However, I'm facing a lot of difficulties and challenges. For example, if we grow, sorry.
I think this is what it's like to load some of these images that you're taking.
I see. Okay. So, okay, thank you. So, if we grow the prostate cancer cells on the dish, these cancer cells usually carry a lot of mutations and high levels of aneuploidy. And these mutations and aneuploidies can also evolve over time, which makes the studies very difficult and inaccurate. And if we grow the normal cells under the dish, these cells are usually grow very slowly, and then they will enter the senescence state, which is a state in which the cells stop dividing. And more importantly, the cells growing in the 2D cultures in this way will lack of many cellular signatures of prostate tissues. So to overcome these difficulties, I collaborated with Dr. Smith from the Vanderheiden lab from our Koch Institute and the Lee lab from the Brigham and Women's Hospital. And we isolated the primary prostate from desired mouse models and developed them into a multicellular organoids, which we can grow them as a many prostate organs in the cellular matrix, which we call the matrigel. As you can see here, by supplementing these organoid growth with a lot of growth factors essential for prostate tissues, they can grow pretty well in the matrigel, as shown here in the light microscope. And then in the stunning pictures that I'm showing, which didn't show up here for some reason, hold on. I use immunofluorescent staining and confocal microscopy and show that these organoids are actually actively growing by staining this Ki67 protein, which marks the cell cycle. And also these organoids expresses one of the very important prostate signature protein called estrogen receptor. And more importantly, I sequenced these organoids over time, and I found that they always showed a euploidy, which indicates these organoids are having a very stable and balanced genomes. So with these organoid models, I can introduce multiple genetic perturbations based on mutations and copy number changes in from the cancer cases. For example, here I introduced the temporal fusion, which occupies 50% of the prostate cancer cases. And as you can see here, it causes some levels of morphological changes. And then we can do a lot of assessments to these organoids, such as evaluates growth and invasive behaviors and etc. And one of the most recent example is that, we just need to let it show up and over some time, is that we, should be fine. Okay. We identify some aneuploidy associated gene. One of them is the RAD21 gene, but when gain of a copy can alleviate some of the cell death effects caused by the oncogene expressing or in the organoids, which in this way, it promotes the fitness and improve the fitness and the growth of the organoids. And then moreover, through a bridge project between MIT and Harvard School of Public Health, we are currently collaboratively investigating the hits that we identify in our organoid studies and see if they have prognostic predictions in the epidemiology studies for the patient cohorts with prostate cancers. And then we also identify some of the hits from the epidemiology study that we can bring it back to our organoid cellular studies to study the mechanistic aspects of these hits. So by these studies, we hope to identify some of the novel aneuploidy associated markers in prostate cancers that may have some implications for therapeutics. So with that, I would like to thank my advisors, Drs. Angelica Among and Dr. Jackie Lees, and all my collaborators and all the institutes and funding resources, especially the Bridge Project, which sponsored my research. And thank you so much.
Thank you very much. So that Bridge Project is actually going to be my transition into the next project. This is another project that is a collaboration between Koch Institute researchers and some clinical investigators. So Megan is going to come and talk to us about a different type of cancer, young at heart.
All right, well, hello, everyone. I am thrilled to present our image tonight on behalf of my team called Young at Heart, Progenitor T Cells Drive Immunotherapy Response. So, all right, good trend. Yeah, so this image is really the product of a fantastic collaborative effort supported by funding from the Bridge Project. So this Bridge Project supports collaborations between researchers at the Koch Institute and researchers over at the Dana-Farber Harvard Cancer Center. So on the MIT side, in the JAX lab, we're interested in using genetically engineered mouse models of cancer to really understand how cancer develops with the goal of identifying new targets for cancer therapies. And we're particularly interested in understanding how to activate the immune system to target cancer cells. And we've teamed up with Sandra Santagata's lab at Harvard to apply their really cutting edge multiplexed immunofluorescence imaging platforms to take a deep look at interactions between tumor cells and the immune system through integration of molecular and spatial characterization of single cells in lung cancer. So CD8 T cells of your immune system are able to recognize and kill cancer cells, but they fail to do this in patients that present with cancer in the clinic. However, new checkpoint blockade immunotherapies are able to reactivate T cell responses. And this has led to really successful responses at controlling cancer across a subset of patients. This can, in some cases, lead to curative responses. However, unfortunately, the majority of patients still do not respond to these therapies. And so the goal of the JAX lab and Sonic Auto lab is really to understand why this is and what we can do to extend the benefit of these therapies to more patients. So recent studies have actually identified a particular type of CD8 T cell that must be present to get a good response to checkpoint blockade immunotherapy. So these so-called progenitor cells have stem-like characteristics, and in response to immunotherapy, they can generate killer CD8 T cells that are able to control the tumor. So these progenitor cells are marked by a protein called TCF1, and TCF1 is actually being considered as a potential biomarker in the clinic to predict patient outcome in response to immunotherapy. So if you have these progenitor TCF1 positive T cells, you're more likely to respond to checkpoint blockade immunotherapy than patients that don't have these cells. However, interestingly, our recent studies in the mice have uncovered that this progenitor CD8 T cell population does not necessarily have equivalent function. So there's actually a subset of these progenitor cells is marked by another protein called CCR6 that does not give rise to killer CD8 T cells in response to immunotherapy. And hence these cells actually don't contribute well to immunotherapy response. And it turns out that these cells are not only found in mouse, but in human as well. And this is the finding that we decided to highlight in our image in the gallery. So we performed multiplex immunofluorescence imaging, where using 3 markers, which I might be able to point to, CD8, CCR6, and TCF1, we were able to identify this subset of dysfunctional progenitor cells across most of the patient samples that we analyzed. And in further work, we were able to show in melanoma patients treated with checkpoint blockade immunotherapy that these CCR6 positive cells don't predict good response to immunotherapy in patients. And so what we hope is CCR6 could serve as an additional biomarker in the clinic in combination with TCF1 to more stringently stratify patients and to better predict which patients are going to respond to immunotherapy. And in some additional work, actually, that we're very excited about, we've also identified therapeutic vaccination as a potential approach to eliminate these CCR6 positive progenitor cells, really enriching for the functional progenitor cell population. And so we're interested in understanding how we can optimally combine therapeutic vaccination with checkpoint blockade immunotherapy to really exploit this progenitor cell population and get a greater degree of tumor control. So with that, I'll just acknowledge the 2 teams again, and I'd like to give a big shout out to Claire Rich, who actually created this gorgeous image that we have in the gallery. I'd also like to thank the core facilities at the Koch for their help with this project and their funding sources as well. Thank you.
Thank you so much, Megan. All right, and Megan has done a lovely job of transitioning us into the world of immune and now immune engineering. So she mentioned vaccines. There are several different types of vaccines, as I'm sure people know, and our next project is going to talk about some strategies for vaccination. So Brittany, if you want to come up and take us behind the mucus of your vaccine breakthroughs.
All right, thanks, Erica. My name is Brittany Hartwell, and I'm very excited to be here with you all today to share some about this image that I collected as a postdoc in Daryl Urban's lab, along with Jason Chang, another postdoc in the Urban lab who's not able to be here today. In the Urban lab, we work on developing vaccines and immunotherapies that can train the immune system to fight disease, ranging from cancer to COVID. This particular image shows a vaccine that we engineered to be taken up across mucosal tissues in the nose in order to activate a stronger immune response to protect against infectious diseases. So why might we want to engineer vaccines that can target mucosal tissues like this? Well, it's because intuitively we know that most pathogens are transmitted by mucosal routes. Take SARS-CoV-2, for instance, the virus that causes COVID. It's transmitted via the upper and lower respiratory tract. HIV is transmitted via the genitourinary tract. So it makes sense that an ideal vaccine would establish immune protection at this mucosal tissue as a front line of defense. But now consider most of the vaccines that we receive when we go to the clinic, they're injected. And this sort of traditional injected vaccine, while very good at establishing immune protection to protect us from severe disease and severe infection, they actually aren't really designed to establish immunity in the mucosa. They establish what we could refer to as a backup defense in the blood. So what we wanted to do was engineer a vaccine that could establish this frontline defense. And one way to do this is to directly administer vaccines to mucosal tissues. All right, so if this strategy of mucosal immunization can establish this enhanced immune protection, why can't we receive, say, an intranasal vaccine every time we go to the clinic? Ultimately, it comes down to a drug delivery problem. Our mucosa is designed to keep things out. So if we were delivering a vaccine by this route, ultimately we'd want to get that vaccine through these mucosal barriers into the immune cells associated with that mucosa that underlie all those barriers. In the nose, this is called the nasal associated lymphatic tissue, or the NALT. And these immune cells are really where antigen-specific immune responses against invading pathogens are orchestrated. But as you can see here, in order to get vaccine components to that NALT, first they have to overcome the mucosal barrier. Typically, antigen components are broken down or cleared away in the mucus. If they get past that barrier, then they're faced with the epithelium and tight junctions between these cells. Ultimately, this is designed to keep stuff out. But we know that a few things can get through. And one of those things is called albumin. It's a naturally occurring protein that's found in mucosal secretions. And albumin is able to get up across these barriers by binding to a receptor that then shuttles it up across the epithelium and kicks it out on the bottom underneath these other cellular layers near the NALT. So we thought, what if we engineer a vaccine that can bind onto albumin, using it as a chaperone to effectively hitchhike using this receptor to get up across these barriers. And by doing that, we could concentrate our vaccine, bypass the barriers, and concentrate our vaccine where those important immune cells are located. And when our vaccine can interface with those immune cells in the presence of antigen from our vaccine, those immune cells undergo multiple rounds of crosstalk, leading to activation of important immune cell players, which can then actually pass back out, migrate into the mucosal tissues where they're established as sentinels or guards to protect against invaders like viruses. And when they encounter those viruses in the mucosa, they secrete protective immune proteins called antibodies that can then bind onto that virus, preventing it from infecting those cells in the first place. And that is actually what we're looking at in this image that's shown in the gallery. We took this image because we wanted to see how our vaccine was behaving after it was delivered intranasally. This is showing a cross-section from a mouse's nose. The mouse was immunized with our vaccine, and 24 hours later, we took this image. The vaccine is showing up here in yellow and pink. All the cells in the mucosal tissue are shown in blue, and the airway or the negative space is shown in black. And the first thing to note here is that this image, again, was taken 24 hours after the vaccine was administered. And we can see a lot of vaccine lining the airway and being taken up into that mucosal epithelium, which indicates that first, our vaccine bypassed that initial barrier of mucosa. It's mucus. It's evaded clearance and degradation by the mucus. And here we see it actually being taken up, binding and being taken up into those mucosal epithelial cells. And if we look closer, we can see it being taken even deeper in those little vesicles and bubbles, the yellow and pink, into those underlying submucosal cell layers as well. And the reason this image is so important is because it shows that our vaccine is able to overcome these barriers for delivery, which ultimately allows it to activate better immune protection at the front line. This is another way of visualizing this. In this case, the cross-section is shown with cells in blue, and the vaccine is shown in red. And we can see zoomed in on those epithelial layers, the vaccine layering the epithelium, being taken up in vesicles underneath. In contrast, if we compare this to a vaccine that lacks this unique engineered property of albumin binding, we don't see any vaccine retained in the airway, on the epithelium, or in the underlying cell layers. But engineering a vaccine that can get across these barriers leads to it trafficking into the nasal associated lymphatic tissue, where it interfaces with immune cells, can activate those immune cells to then go back out, into the mucosal tissues in order to establish mucosal immunity in, for example, the nose, the lungs, or the genitourinary tract. And our research with this vaccine platform so far has shown some really promising results, showing that we can establish functional immune protection against HIV, for example, in the genitourinary tract, or against SARS-CoV-2 Against SARS-CoV-2, we've shown that we can activate antibodies in the nose and in the lungs that can neutralize virus before it's able to infect cells. And so thank you so much for your time. And I'd also like to thank my postdoc advisor, Daryl Urban, and also Jason Chang, the other postdoc who helped collect this work, and a few of the cores here at the Koch Institute who helped in the collection of this image, especially the histology facility in Kathy Cormier. Thanks so much.
Thank you, Brittany. So given the immune response that you orchestrated, I am going to stick with the music puns and say that it's sure to be a chart-topping albumin. All right, our final image of the evening also deals with vaccine delivery. And here we are. So I'm going to invite Dhruv up to talk to us about this beautiful array of light.
Hi, everyone. My name is Dhruv Varshney, and I am a technical associate in the clinic group of the Langer Lab. And today we will be presenting over our array of light, fine-tuning the production of 3D printed microneedles. In this image, you see 3D printed microneedles from the highest resolution 3D printer in the world, the Nanoscribe. We use these 3D printed microneedles to develop mRNA vaccines against SARS-CoV-2. As we all know, in the last 2 to 3 years, mRNA vaccines have dramatically improved vaccination efficacy, specifically against SARS-CoV-2, but they have also changed the landscape of immunology and vaccinology for the next generation of vaccines. In mRNA vaccines, there are 2 main components, mRNA, as I hope that was obvious, and also LNPs, or lipid nanoparticles. LNPs in mRNA vaccines are composed of 3 crucial elements, ionizable lipids, helper lipids, and PEG lipids. And together, these simultaneously preserve and serve as stabilizing excipients for the coding sequence of the mRNA so that the vaccine is delivered to the appropriate spot in the body for the highest possible immune response. As such, the platform is highly immunogenic and also incredibly modular, as in you can plug and play mRNA into virtually any disease model using the same combination of stabilizing excipients to target virtually any disease. However, there are still many issues with this platform. The generation of biohazardous sharp waste, the thermostability of the vaccine itself, mRNA needs to be kept extremely cold, the need for healthcare personnel to actually administer the vaccines, and also the pain that is associated with vaccination, which can be a deterrent for people to choose to get vaccinated. As such, there are biomedical alternatives for standard of care vaccination. One of these are microneedle array patches, or MAPs. MAPs are painless, thermostable, and sharps-free biomedical alternatives for vaccination. There we go. So the way that MAPs work is that they are able to spatially control a vaccination to the dermis, or the layer that is about 1 to 4 millimeters from the very top of your skin. The reason that the dermis is targeted is because it has a very high population of resident immune cells, including T cells, B cells, mast cells, and dendritic cells, just to name a few. And when targeted, these cells are able to take an antigen that is presented to them and create a very robust immune response. And so you can imagine that the actual design, the geometrical design of these MAPs is very crucial to ensuring the highest possible vaccination. Some of these considerations include pitch, or how far apart the needles are spaced. Having a needle, having a MAP that is not spaced appropriately could lead to the MAP just popping out of your skin after application. The patch size, or how large your MAP actually is. The reason that patch size can vary is due to the potency that's associated with the vaccine, or the actual dose that is required for generating an appropriate immune response, and also the tip angle, or the sharpness of the vaccine. Having a very sharp microneedle is crucial to targeting the dermis accurately, otherwise you'll be getting a suboptimal response. And so you can wonder, or you can imagine rather, that being able to simultaneously and rapidly test these factors to create the most optimized microneedle array patch is very crucial to advancing this platform. So to meet that goal, we use MPP, or multi-photon 3D printing, to enable the rapid prototyping of optimized maps for enhanced vaccination. There's a very simple workflow associated with this process. First, we create a computerized assisted design, or drawing, using common software such as SolidWorks or AutoCAD to actually design our microneedles. Then we think about the actual material that we're going to print out of, including the rigidity, the internal structure of our needles, the size, and the desired accuracy of our features. We then encode these parameters into a general writing language in which we tell the nanoscribe this is exactly what we want. Following this process and this encoding, we physically print our needles and develop them. Development is a process in which we take the unpolymerized material and remove it using a chemical process. Following this, we are able to characterize these beautiful needles, create a reverse mold, and then cast dissolvable polymers, as well as mRNA, which we then use to test or assess the immunogenicity of our vaccine in animal models and hopefully in the clinic soon. And so I'd like to thank lots of people that work on this project and our collaborators, but primarily Anna and Bob for all their advice on this platform and also several other projects in the lab, as well as the Bill and Melinda Gates Foundation for funding this project and the rest of the Clinic group members. And I also actually have the award-winning image in my hand. If anyone wants to see it, I can pass it around.
Thank you, Drew. Zoomers, we will make sure to get a picture of this and put it on our brand new Image Awards archive website with this image. So thank you so much, Drew. We only have 85 words or so in the caption, but I like that these presentations can take us point by point through everything. All right. One of the other things we liked about this image was the reflective surface on the silicone chip here. And so that is going to be my transition to a closing reflection from our director, Matt Vanderheiden.
Thanks so much, Erica. I'm not going to try to follow up those puns. You guys will be glad to know. I just want to start by thanking everybody for joining us, both here in the Luria Auditorium and on Zoom. The KI Image Awards really is like a highlight for me and for many of us here at the Koch Institute. Kind of signals the beginning of spring. It's a great way to highlight the wonderful work that we do here. But this year it's a little bit extra special because this is the first in-person event that we've been able to have in 2 years. And I hope that this is just the first of many as we finally reach a new stage in the pandemic and hopefully return to a much more normal way of doing things. I want to personally congratulate all of the winners. I was really excited and inspired by your talks. I'm sure I'm sharing sentiments that are shared by everybody here. I think it's particularly cool how they really highlight the mission of the Koch and what we are trying to do here. There is great examples of engineering. There's great examples of basic discovery, and the stories behind those images, apart from the images themselves being beautiful, I think really highlights the difference that what we're doing across the entire spectrum to hopefully change the reality of how cancer is treated and really many different other types of diseases. I think it's also particularly cool that it's our trainees who get to give this talk. It's obviously incredibly important for us here to train the next generation of cancer scientists. Those who know me well know how much I value the trainees who are really the heart and soul of what we do here. We would not be able to do the amazing work we do without you guys and your contributions. And I think it's wonderful that you're able to participate in this and really share with us the real stories behind how the discoveries are made and not just the ones that us in the PI chair sometimes like to tell. So I also want to acknowledge that today is MIT's 24-hour Giving Challenge. I know many of you here have participated in that. I want to personally thank you for that. And I also want to highlight that we have a new website here at the Koch Institute. So if you haven't checked that out, please check it out. And you can learn not just about the projects that you're here today, look at past stories of image awards, past beautiful images that have come before the winners today, but also learn more about the huge range of research that we do here at the Koch Institute. So just with that, I know the hour is getting late. We have a reception afterwards. I want to invite everybody who can to please spend some time with us, get to know some of the awardees, enjoy the images out in the in the gallery. And it's really wonderful to see all of you, and thank you again for coming. That's okay, I don't have to switch.
All right, thank you. And I'll invite everybody on Zoom, please come visit us at the Koch Institute and come see these images. All right, thank you all. Have a wonderful night.